In this session, we'll learn how to translate nucleic acid sequence into a protein sequence. The first step is to learn how to navigate the RefSeq protein sequence as well as the coding sequence within the mRNA RefSeq entry. In the mRNA RefSeq, you'll find the coding or CDS link and then note the position of the A in the ATG start codon. So let's go back to the NCBI gene reference and then pick CFTR again, and we'll find the entry that we've worked with in an earlier session. We'll click that and it will bring up the RefSeq entry for CFTR. So as that loads, we'll use the link on the right and we'll go to the NCBI RefSeq entry. So this time we're going to note the RefSeq protein entry, and we'll go ahead and click on that, and it will open a file that will show us the full protein sequence for the CFTR protein. So we'll click on that and load the CFTR protein RefSeq. And again, you can see that it has a unique identifier. Then we can scroll down into this file and see the full protein sequence for the CFTR protein. So this will be an important point of reference as we review our work further into the session. So now let's go back into the RefSeq mRNA entry and we'll click on that link. And this time we'll scroll down into the file. We're looking for an entry referenced with the three letters CDS. This will tell us the coding sequence range for the CFTR gene. So once we find that, we notice a few things about it. It gives us some position. So it says here 133 is the starting position of the coding sequence. We can actually click on that link and it'll take us down into this gene sequence and highlight that region. And it also brings up a pop-up window displaying the translated protein product. We can use all of this information to validate our work using the Bioinformatics platform. Now let's learn how to use a bioinformatics application, SeekBuilder by DNA Star, to convert nucleic acid sequence into a protein sequence. Within SeekBuilder, there are a few configuration settings that you'll want to make. Under the ORF settings, under the sequence menu, make sure that you choose the only show ORFs with a start codon option, and that you set the minimum length to 25 amino acids. Once you do that, then you'll open the application and find the appropriate ORF or open reading frame. There are a few clues that you'll use: the length of the product, the start location compared to RefSeq. Another clue is that RefSeq will not use the opposite strand reading frames numbered 4 through 6 in SeekBuilder. Then you'll convert the ORF to a protein sequence and again compare it to known sequences to verify your work. So now let's go back to SeekBuilder, and again we're going to open our CFTR sequence as we have in earlier sessions, and then that will install into the application. We change our view from RNA to DNA for simplicity. And then now we're also going to need to change the open reading frame parameters. And so we'll pick this option and we'll make sure that we have a minimum of 25 amino acids in any reading frame, and we'll change that to only include sequences with the start codon. And then under this menu we can expand and see the open reading frames. There are 6 possible open reading frames for any DNA sequence. 3 for the top strand and 3 for the opposite strand. And you can display or hide using the plus or minus checks as I just showed you. So let's look at the position for this particular one. Remember earlier we saw that our sequence starts at position 133. So the open reading frame number 1 in red is most likely to be the open reading frame that we will accept for CFTR. So let's take a closer look. Let's translate it. And again, by default, it shows all 6 possible translations. So let's clean up our view a little bit and remove the bottom strand. And then let's narrow it down so that we remove open reading frames 2 and 3, leaving us only the strand for our predicted protein sequence. In bioinformatics, it's always important to verify your work using whatever resources possible. So to wrap up this exercise, let's go back to the RefSeq view, and we can see the predicted protein sequence that we identified from RefSeq. And then on the right, we can see the sequence that we predicted using SeekBuilder. And if you just read through those, you'll see that they are indeed the same sequence, helping us feel confident that we have correctly translated the nucleic acid sequence into a protein sequence. In this session, we've learned how to navigate the RefSeq information as it pertains to translating nucleic acid sequence into protein. We've also learned how to use a bioinformatics application to identify an open reading frame and translate that sequence into a protein sequence.